So as one of the non-scientists in the room, I thought I would start with the role of FDA as compared to NIH. I know I only have 5 minutes, so it's not going to be a long role. But so in this space, you've got NIH doing this super cool research that is leading to amazing discovery that helps us better understand the molecular basis of disease. And then it shifts over with industry's help to the FDA, and we translate that discovery into these precision cures, which is pretty amazing. So what we do on our side is we take the data that's generated by NIH, by industry and others. We take a look at it, and we understand then that there are these targeted therapeutics that actually only work on certain subpopulations of people with a given disease. Right? So they only work because we understand that this person presents not just with this disease, but with this disease that is accompanied with a specific genetic variant. And so that is precision medicine. That's what, and in the field of cancer research, it's really taking off. It's really amazing how much we're understanding. What we're understanding now is that breast cancer and ovarian cancer don't necessarily have to be treated as a different cancer with different therapeutics, but they actually may present with similar genetic variants that may be responsive to the same type of therapeutic. So that gets me to today, which is the diagnostic piece, which is why it's so important that the diagnostic accompanying the therapy, be be accurate. Right? I mean, if it's not accurate, if it doesn't identify the right people, we're not going to have the right result. So it's not going to be terribly precise. So where we are today, we've had a number of targeted therapies. I was reading some literature as I was preparing for this that said it all started with Herceptin, and understanding that in certain breast cancers, they presented with what was called the HER2 protein, and and Herceptin was designed, and we understood that it wasn't going to be effective on all women with breast cancer, but only a subpopulation that presented with this HER2 protein. And so when Herceptin came to the market, they had to have what was called a companion diagnostic. And the FDA has issued guidance on this, that when the drug company that is developing a targeted therapy, when they come to the FDA, they have to come not only with their drug, but with the diagnostic they intend to use to identify that subpopulation, because it's extraordinarily important that FDA not only be able to evaluate the data that's submitted to establish the effectiveness of the drug and the safety of the drug, but also the accuracy of the diagnostic tool that's used to identify the population who would benefit from that drug. So we've approved over 20 companion diagnostics. We do it under, I don't know how much you know about the FDA, but we have PDUFA guidelines, PDUFA deadlines, right? So that prescription drugs, when a company submits an application to the FDA, we have certain deadlines we have to meet to approve that or to make a decision on that product. And so they bring in not just, again, the drug, but the diagnostic, and we approve both within the same time frame. So we're talking about ovarian cancer, breast cancer, cystic fibrosis, colorectal cancer, melanoma. There are a number of targeted therapies right now, and the list is just going to grow exponentially. So it really is an exciting time for precision medicine. So that's sort of where we are now. And the common theme with those diagnostic tools, those companion diagnostics, is they're built to look for a specific genetic variant. And so when they come to the FDA for approval, it sort of fits with what we've done before. Like, we look at diagnostics that are intended for a single purpose, right? Or are there to look at to identify a specific group of things that matter. And they sort of fit within our current paradigm of, we have these really excellent scientist reviewers who will make the test sponsor run their tests, who knows how many times, to make sure that they always perform the same way, that they accurately identify what they're supposed to identify. And we have confidence that when these are used on people, they're going to get a good result. So that's fits with what we do. So then comes precision medicine on the president's precision medicine initiative. And for us, what that means is next generation sequencing. And when I learned as a non-scientist, what that means is a test that can actually sequence the entire genome, your entire genome. And that sounded big, but sort of like I couldn't tell how big, but then it's billions of base pairs. So imagine FDA trying to make sure that every one of these billions of base pairs are accurately measured. Like we would just keep on testing into the, forever. So it's absurd that we would ask a company to to present us with data that every billionth one would be measured accurately. But what's good enough, right? There's millions, each of us, I learned this too. We have millions of genetic variants, all of us. They just don't all necessarily translate into a disease. But we all have variants. We are not orderly people. We all have these variants within us. And what I learned is, and our DNA doesn't behave like it's not orderly. You would think it's like, you see the letters, the 4 letters, and you think that they're gonna be lined up in a row and they're gonna behave all that way down down your DNA, down the genome. That's so wrong. That is completely wrong. There's parts that duplicate each other. There's huge gaps that are missing. There's things that are sort of mirror opposites of what they should be. And so all of that makes the FDA really worried about, is this test going to work? And how do we demonstrate that your next generation sequencing tool is actually going to perform right? Because it's not just measuring a very orderly line of these base pairs, as they call them. It's like measuring all of this weird stuff that happens along and knowing that it's going to be able to detect it correctly every time it's run. So what we did with the cystic fibrosis platform, which was called the MySeq DX, this was our first next generation sequencing tool that we used. And so we had the platform come in, and what we asked of them was, we have these great, geneticists on our staff who understand the parts of the genome that aren't orderly and don't behave right. And they made them test that part, like all the different parts that don't behave right, and see what they found. And could they consistently find the same thing that we knew, we would know the answer, right? Can you consistently test it, and does your answer come out like it's supposed to? On these really weird flipped, breaks in data, all those kinds of things, do you measure it right? And so that let us get down from 1 billion to a much more manageable number. So again, in an agreement with the sponsor, we had them present data that they could accurately measure consistently all these different parts of the cystic fibrosis gene. And based on that, we cleared the platform. And based on the work that they did, we were able to develop standards for other platforms so that they could show us that they met the same standards that this early tool came on. And they didn't have to come before the FDA at all. We could just approve them because we knew that they would also function well. They would also be accurate every time they ran their test because they had the same tools in place that the original tool had. So that's sort of the paradigm. So now we're shifting from the cystic fibrosis gene to Kathy's world, where they're going to map the whole DNA. They're mapping everything. We want that research to be right. And so how do we make sure that it's right? And how do we make sure that these genetic tests are right when it is not just we want to diagnose a disease, but we also want to be able to predict whether you might be at risk of a disease, which is really tricky because that risk factor may be that much. And so what's tricky is that risk projection accurate? Is what they've measured accurate? And then the second challenge for us is how meaningful is it? So the challenge that we have with next generation sequencing comes to 2 things: our role with the president's, the PMI, President's Personalized Precision Medicine Initiative. We have to do that accuracy part. It's called analytical validity. And what we've been charged to do is to find a way to make sure that these tests are accurate, but not take too long to do it so that we stifle innovation. So it's trickier than it sounds. But what we've done, again, is we've learned from what we did with the cystic fibrosis tool. And what we're doing now is we've partnered with NIST as well, which came up with this very cool thing called Genome in a Bottle. It's actually called, it's reference material. And what it is a tool that labs can use because we know where the variants are in this genome in a bottle. And so the labs who are developing tests can run their test on the genome in a bottle and see if they get the same result as they should. Like, did they find the variant they should have found? Did they not find a variant where there shouldn't have been a variant? And so it's sort of like you have the answer book in the back of your book, right? Like, I can test, did I get it right or not? It's yet another tool that we're working to help labs have so that they will know themselves whether these tests are accurate. And then the other thing that we're doing, kind of on the same line, one thing about much of the genome is that it is actually sort of well understood. It does sort of behave similarly. Like, when you have that kind of massive data, you also end up having, being able to see that it has properties, it behaves a certain way, and you can actually develop software algorithms that sort of check, like, whether this test result makes any sense given what we know about it. It doesn't work for, like, the cancer part of the genome, which is sort of all mystifying and kind of still very not well understood, but it does work for a big part of the gene. So we're looking at developing sort of a software checking tool to help people, again, check for themselves, did my test run correctly? And then finally, what we're doing with NIST and the scientific community on the analytical validity side is we're exploring whether there are standards that could be agreed upon that would ensure analytical validity of these tests, these diagnostics. And the goal would be if we could have confidence based on this kind of standards approach, which is beyond my level of scientific understanding. I'm not really exactly sure what they would look like. But if we have that type of confidence, we would alleviate the need for all of these tests to come into the FDA. We might be able to be confident that they'd be accurate, and we'd be able to assure the public that they should have confidence in the test results. So that's the one side. That's what success looks like there, is are we able to design a regulatory system that assures accuracy of these tests so that you all can be confident that the test results you get are accurate consistently and do it in a streamlined way. And the second major challenge that we have is what's called clinical validity. So now we know that your test accurately measures this one piece of the gene. What does it mean? Is it relevant? Is that genetic variant relevant at all to a disease? And I never thought about this before the Precision Medicine Initiative, but I've given a lot of thought about it. So we're at this exciting time in precision medicine because it's built on decades of research. There are decades of discovery that are stored in databases across the country, different labs, different academic centers, industry. They've all been seeing patients and better understanding people who have these variants and who have a disease and linking one to the other, one to the condition. And all of that data is stored not in one great place in a well-curated database like the FDA would love to see, but in disparate places, sometimes like with, using different methods so that they don't make sense with each other. And we don't really know, like, how robust is the evidence out there that says if you have this genetic variant, you are either going to have this disease, likely to have, you have a risk factor, it is predictive of, or it can be used as a diagnostic. So we don't really have that, except in certain instances. Like the Cystic Fibrosis Foundation, they partnered with Johns Hopkins. They have an excellent database that's allowed us to approve certain tests so that the company isn't going to have to come to FDA saying, here's our test that measures the entire genome, and here's our literature that supports what it means. Because to have like each company come to us and try to demonstrate what, the connection between the variants they want to claim they measure and the disease that it's linked to, it wouldn't make sense for any of us. It would be a one-off approach. Everyone would be coming to the FDA to demonstrate the same thing. And wouldn't it be great if we could instead have this community-based community-generated evidence in a well-curated, we're calling it a regulatory grade database, which basically means that you have the same smart scientists who are like at NIH and academic centers across the country who are making sense of discovery in a way that we can all understand. So that's the other part of success from the FDA perspective. Thank you.